Okay, welcome back. Let's take a look at our concept questions and then we'll get on with translation. So what region identifies the start of the gene to the RNA polymerase? Well, that would be the promoter region, because that's the region of the DNA that the RNA polymerase is going to bind to. So that identifies the beginning of the gene. In eukaryotes, the final mRNA is shorter than the original transcription product. Why? Well, we were just talking about that. Remember? In eukaryotic genes, there are both introns and exons. Non-coding regions, those would be the introns, and coding regions, those would be the exons. And to create the final messenger RNA, you need to cut out the introns and splice together the exons, which automatically means that the final messenger RNA will be shorter than the transcript because you've cut part of it out in order to splice together the remaining bits to create the messenger RNA. Not difficult ideas to process, I think. Anyway, let's start talking about translation. So in translation, we are obviously changing from nucleic acid and its language to proteins and their language, hence transcription. It turns out that there are multiple players in this process. So let's take a look at the players. The first you've already met, that's the messenger RNA. That contains the sequence of nucleotides that are going to be divided up into codons and read off to allow the assembly of the coded amino acids into a polypeptide. But the other two players that are involved are the ribosomes, which actually carry out the assembly of the polypeptide, and then a second RNA molecule called transfer RNA or tRNA, which is the actual cellular interpreter. So let me put this here. Because whenever you do translation, you need someone who speaks both languages and can therefore do the interpretation from one to the other. And in cells, it's the tRNA. It speaks the language of nucleotides because it is an RNA molecule, so it's built out of nucleotides. But it also speaks the language of amino acids. And when we take a closer look at tRNAs, you'll understand why. So let's take a look at the tRNA in detail. So here is a pseudo 3D model of the tRNA. It actually folds up into a three-dimensional structure. A more simpler way of drawing it, and a way that has been classically used, is to draw it like this. And this is called the hairpin loop structure. Which is how it actually looks in pseudo 3 dimensions in this diagram. Where the anticodon is located is right here in the center of this loop. That's where the anticodon is located. So the three nucleotides of the anticodon are located here. And what does the anticodon do? It pairs with the codon in the mRNA. So how does this speak amino acids? Well, the pairing of the anticodon with the codon is the key, because whatever codon the anticodon pairs with determines what amino acid is going to be attached up here at the amino acid attachment site. So the anticodon and the codon that it pairs with determines the amino acid. So this is how it can speak both nucleotides and RNA. It speaks nucleotides through its anticodon, and it speaks amino acids through its amino acid attachment site. Remember again, the anticodon base pairs to a codon on the RNA. That codon codes for a particular amino acid. That is the amino acid that's attached to this tRNA. So that's how it can interpret between nucleotides and amino acids. So this is all just documenting that. The other player is the ribosomes. And the ribosomes are made up of two particles, what are referred to as a small subunit and a large subunit. Within those subunits, there are RNA molecules and proteins. And it turns out that we now recognize that the RNA molecules are actually the key players here. They are the enzymes, we refer to them as ribozymes, that link the amino acids together. Oh, it's so untidy, let's clean that up. So we now know of a broad range of these RNA molecules that have enzyme function. So as much as we identified enzymes back in the previous chapter as being proteins, we do now recognize that there are also RNA molecules that also have enzyme function. We refer to those as ribozymes. And here we encounter them in the ribosomes, acting as enzymes to link the amino acids together. And in fact, this is where the ribozyme concept first came from, hence the name. Not terribly important that you know that, but again, some information that I think is important for you to know. I would like you to know it, even though it's not necessarily mentioned in the book. So when we take a look at the ribosome, we recognize a number of different sites. Now, the book shows two. We actually know that there is a third site, and it is located here, and it's referred to as the E site, and that is for exit, because it is through the E site that the tRNAs exit the ribosome when their job is done. The P site always contains a tRNA that has the polypeptide attached to it. The A site always has the tRNA with the next amino acid to be added to the polypeptide attached to it. So P for polypeptide, A for amino acid. That's the easiest way to remember it, and of course, E for exit. Then you have the large subunit, which is the enzyme site or the catalytic site, the small subunit, which serves as a regulator and also helps clamp the messenger RNA into the ribosome so that it can feed through the ribosome and feed through the sites. Then at the base of each of the P site and A site, there are going to be codons. The codon at the base of the P site base pairs to the last tRNA that brought an amino acid with it and therefore has the polypeptide on it. The A site has the codon for the next amino acid to be added and therefore has the tRNA with that amino acid on it sitting attached to it. So here you can see the artist representation of how these three players interact with each other: the messenger RNA, the ribosome, and the tRNAs. So let's take a look at the steps of translation, of which there are three. So they break up into initiation, elongation, and termination. And it's really important that you recognize that these three steps of transcription and translation, while they are named the same, are very different, and that neither of them have anything to do with DNA replication. This is something that I see students doing over and over again, confusing the steps of translation with the steps of transcription, because they're named the same, but what happens in each of them is very, very different. And then thinking that there is somehow a connection between transcription and DNA replication. They are very different processes. Make sure that you understand quite clearly the distinction between all three processes, because they are distinctly different processes. There are three distinctly different processes. Really important that you recognize that. So let's take a look at initiation first. In initiation, you've got to bring together the messenger RNA and the two components, the two subunits of the ribosome, along with the tRNA that is going to bind to the start codon, which is, of course, a tRNA for the amino acid methionine, because it serves the same purpose. So remember, we have a cap at one end of the messenger RNA and a tail at the other end. Well, the cap is at the 5' end, and that is the starting end of the messenger RNA. Messenger RNA is always read 3' to 5'. So mRNA is read 5' to 3'. So this is the 5' end, and this will be the 3' end. It is the presence of the cap and this short sequence of nucleotides that identify this as the start point for the gene, for the ribosome. So once it identifies that this is the starting point for the gene, the small subunit attaches at this point, locating itself adjacent to the start codon. The tRNA for the methionine then attaches to the start codon. And then the large subunit attaches and positions itself so that the start codon and the tRNA for the methionine occupy the P site. Remember, P is for polypeptide. And right now, the polypeptide consists of just that methionine attached to the tRNA bound to the start codon. But what that does is position everything into context. And it establishes what is referred to as the reading frame. So it establishes how you're going to read off the triplets or codons. We say that it establishes the reading frame. So obviously getting the ribosome correctly oriented to the start codon is critical to being able to get the right polypeptide generated from the messenger RNA. So this animation walks you through how that takes place. It gives you what happens in translation. So this shows you the basic process of translation. So it's a lot simpler than the BioFlex, so it might be a little bit easier for you to understand. So you might want to take a look at this one as well. But this is basically what's happening. The small subunit aligns itself with the start codon. The tRNA then binds to the start codon, which brings with it a methionine. And then the large subunit orientates itself to that tRNA and positions it in the P site. That leaves the A site open, but this triplet, oops, the pen, this triplet represents the next codon. So in essence, once you establish the start codon and you establish that triplet, then every other triplet is read off as a separate codon. So this is the reason why the initiation step is so critical. If you get the ribosome misaligned, then you're not going to get the right polypeptide. So lining up with the right sequence of nucleotides, the AUG, and positioning the ribosome around that AUG is the key to getting the right polypeptide being produced. Elongation then becomes a cyclical process. And we'll just jump straight to the diagram because it's easier to understand that in the diagram. With a codon sitting at the base of the A site and the tRNA with the polypeptide occupying the P site, the tRNA that base pairs via its anticodon to this codon drops into the A site. That triggers the transfer of the polypeptide from the P site tRNA to the A site tRNA with the formation of a peptide bond between the two amino acids. So the polypeptide has now been transferred across to the tRNA in the A site. This tRNA has finished its job, so it exits from the ribosome through the E site. The ribosome then moves to relocate this tRNA into the P site, which leaves the A site vacant, but with the codon for the next amino acid occupying the base of the A site. So the cycle begins again. And so long as a codon that codes for an amino acid occupies the A site, this cycle will continue. But of course, the question then becomes, we know somewhere along the way, we're going to encounter a stop codon. What happens when a stop codon moves into the A site, which it will inevitably do? Well, that triggers the third part called termination. So when the stop codon occupies the A site, and I don't have a diagram for this, a signal is sent via what is called a release factor. That release factor actually drops into the A site because of the presence of the stop codon. That triggers the release of the polypeptide from the tRNA in the P site. And so the polypeptide is now free and goes off to do its thing. The tRNA, having finished its job, exits through the E site. And with the release factor still sitting in the A site, that triggers the two ribosome subunits to split, release the messenger RNA, and those two subunits then go off to be recycled back into new ribosomes, probably working with other subunits, not working together again. And so that represents the end of translation. Fairly straightforward process. But remember again, different, while the names are the same as the steps in transcription, very, very different processes involved in each of the three steps. So make sure that you are able to distinguish between or differentiate between initiation, elongation, and termination in transcription versus initiation, elongation, and termination in translation. And also make sure that you understand the difference between transcription and DNA replication. I see people getting confused over that all the time. Just make sure that it's not you. So we talk about the flow of information again as DNA to RNA to protein. That's the dogma as developed by Francis Crick. So we can basically draw out a diagram that looks like this. I just want to check this. Yeah, the reason why they're saying that the dogma has been challenged a little bit is because we know that it is possible, and I'll make a note here. It is possible to go RNA to DNA. So some viruses and this process where you go from RNA to DNA is called, and here you can see how imaginative a scientist can be at times, reverse transcription. And the enzyme is called reverse transcriptase. And some cells actually contain a reverse transcriptase, and some viruses actually possess their own. So there are different ways of dealing with this, but all RNA viruses typically have a way of making DNA copies of themselves because it's a good way to either hang out inside a cell in a non-threatening way, which is what HIV does until it morphs back into its dangerous form. It starts making copies of itself. Or is a way of providing a template for making more RNA molecules that become the RNA molecules of the new virus particles. Either way goes. So this is something that's relatively new. We've known about this for about 30 years. So it's relatively new, comparatively speaking. But this is the basic flow of information. So we've got transcription making the copy of the DNA. And of course, this is the eukaryotic model. So in case you are confused, you are not remembering, let me just put this in here. This is the eukaryotic model. So you have the transcript, which then gets processed. It gets capped and tailed. You cut out the introns and splice the exons together. The completed messenger RNA then comes out into the cytoplasm, where it is translated by the ribosomes to assemble the polypeptides. Pretty straightforward. So we'll take one more short break. I'm not going to stop at this point, I think I'm just going to go straight on through into looking at mutations after we answer these concept check questions. These are fairly straightforward. So what is the role of an anticodon in translation? Its role is to base pair or bind to a codon in the messenger RNA and bring with it the amino acid that is coded for by that codon. That's how it functions as the cellular interpreter, which is its role. It is to interpret between the codons of the messenger RNA and the amino acids that need to be assembled into the polypeptide. So that's what it does. These are fill in the blanks, so I think I'm going to fill them in red just to make it a bit easier to see them. So transcription is the synthesis of messenger RNA using DNA as the template. Translation is the synthesis of polypeptides. Probably should have made this a little bit longer. With one codon determining each of the amino acids in the sequence. Okay, let's move on and talk about what happens when things go wrong with transcription and translation. Mutations. So, technically speaking, a mutation is any change in the nucleotide sequence in a gene. That's what constitutes a mutation. Sometimes these can be quite extensive, but the most frequent form of mutation is what is called a base pair mutation or single nucleotide mutation. Now, it's most frequently referred to as a base pair mutation, because if you change the nucleotide in one of the two strands, you are automatically going to change the matching nucleotide in the complementary strand when the DNA is copied. So if you change one nucleotide, you must change its partner as well when the copying process takes place, the replication process takes place. So this is why we refer to them as base pair mutations. So the mutation that gives rise to sickle cell is one of these. It is a single base pair mutation. So here is what the normal sequence of nucleotides would be in the DNA for what is referred to as the beta globin. And this is at position number 6. So this is amino acid number 6 in the sequence of amino acids in that polypeptide. It is a CCT combination, which in the messenger RNA is a GAA. and that is a codon for the amino acid glutamic acid, otherwise known as GLU. So this is glutamic acid. The most common mutation is to replace the T with an A. So instead of being CTT in the DNA, it becomes CAT. In the messenger RNA, it now becomes GUA. C still pairs with G, T still pairs with A, but an A in the DNA pairs with a U in the messenger RNA. So the U replaces any T's that might have been present because the U is the nucleotide that's in the messenger, in the RNA, not the T. That GUA is for the, a codon for the amino acid valine. So you're going from acidic to neutral. And because this amino acid happens to be in a location that is critical for the correct folding of this particular polypeptide, that single change, a single amino acid change as a consequence of a single nucleotide change, means that the protein or the polypeptide no longer folds correctly. And that leads to all the problems associated with sickle cell. What seems like a very simple and perhaps not that serious a change actually turns out to be quite serious, all because of its location. If this nucleotide change was down here in the third nucleotide, it might not make any difference at all. Because remember, we talked about the idea of redundancy and the notion that the third nucleotide is the wobble base, that it can change without changing the overall message that's being sent, because the difference between those multiple codons is at the third nucleotide. So if the mutation had been here, it might have had zero change. And we would call that a silent mutation. Is it a mutation? Yes, because it's a change in the nucleotide sequence. But it's silent because it wouldn't lead to any change in the amino acid sequence in the polypeptide. You'll see what I mean in a moment. So the two most common types of single mutations, or point mutations, to give them their correct name. So single base pair mutations. So there are two ways of achieving a base pair mutation or a point mutation. You can substitute one nucleotide for another. So that would be the sickle cell mutation. Because you changed the existing nucleotide for another nucleotide. That's what we mean by substitution. Or you can remove a nucleotide or add a nucleotide in. And these are the most serious. Why? Because it changes the reading frame. And I can give you an example of this. So here is a message. Now, normally I would hide this in amongst a sequence of letters, but I'm not going to today. I'm just going to make it easy. If we break this up into 3-letter words, it becomes really obvious what the message is. The cat ate the rat. But what if I leave a letter out? Or what if I put an extra letter in? Now when we break these up into 3-letter words, we suddenly wind up with a message that goes from making sense to something that doesn't make sense. And this is the problem with insertion and deletion mutations. These are the most serious because they always make a change. There is no possibility of having an insertion or deletion as a point mutation that will not have a change, will not make a change. Now, sometimes you can actually delete a whole codon or insert a whole codon, but those are very rare because there you're talking about either taking out 3 nucleotides or adding in 3 nucleotides. Very rare situations. Most likely, it is going to be these point mutations, adding in or taking out a single nucleotide. So this diagram simply illustrates the notion of a base substitution versus an insertion or a deletion, but I showed you with the sentences on the previous slide exactly what's going on there. This is taken straight out of the textbook, so you can take a look at this diagram in the textbook and see the same thing. So a missense mutation is one where you change the amino acid, but you only change a single amino acid. So this particular example of a base substitution, let's change this back. And the reason why it's referred to as a missense mutation is because most of the amino acids are present in their correct order. There's just one that has been altered. So this may very well be a fully functional polypeptide. It depends on whether this change has occurred at a significant location. So in the case of the sickle cell, it did happen to occur at a very important location involved in the correct folding of the polypeptide. So even though technically it's a missense mutation, unfortunately, this missense mutation did not result in a fully functional polypeptide. It resulted in a polypeptide that had lost its normal function completely and led to a badly misformed hemoglobin that led to all the other problems with sickle cell. When we change the overall sequence of amino acids, not just a single amino acid, but all the sequence of amino acids, or we prevent the production of a polypeptide, we refer to these as nonsense mutations, because we will likely wind up with a polypeptide that has no function at all because of the significant change in amino acids in the sequence. So we may wind up with a truncated polypeptide if we create a new stop codon. We may wind up with an extended polypeptide because we may lose a stop codon, and who knows where it's going to finish. But what we are going to wind up with is a polypeptide that is almost certainly very, very different from what the original polypeptide should have been. And under those circumstances, it is very, very unlikely that the resulting polypeptide will be functional. You may luck out from time to time, but you can't count on that. It is almost inevitable that you're going to wind up with a non-functional polypeptide. And the closer the insertion or deletion is to the beginning of the polypeptide, the worse the outcome is, because a larger portion of the polypeptide is affected. So this is just going back over all of the things that we've talked about in the case of mutations, so you can read this through from the slides that you can download and get more information, or you can stop it there and get them. The other thing that we need to be aware of is that much of mutations occurs in response to environmental conditions. Now, sometimes mutations occur as a consequence of the copying process. The copying process is very accurate, but it's still not perfect. So replication creates about one error in every 1 billion nucleotides copied. So you can see that it is extremely accurate, but not perfectly accurate. It can make errors, and every one of those errors is a potential mistake or a potential mutation. Where we really run into problems is when we expose ourselves to increased levels of agents that cause mutations, otherwise known as mutagens. So getting X-rays, ultraviolet light from the sun, particularly where our skin is concerned, chemicals like the chemicals in tobacco smoke and other chemicals that can be present in the environment, both natural and man-made. All of these can create mutations, and we have a pretty good handle on what the mutations are because there are only certain types of changes that can actually occur. And so through our investigation of the nature of mutations, looking at how DNA can be altered and also how DNA can be repaired, because there are repair mechanisms that can fix these if the fix can be made before replication takes place. So let's see, cellular repair mechanisms can fix some changes, not all. So once the replication process begins, it's too late to fix whatever change has taken place. Now, not all mutations can be fixed by the repair mechanisms. It's usually physical changes to the DNA, and there are some mutagens that create physical changes to the DNA. Those can typically be identified and repaired. But where you've got a substitution of a nucleotide or an insertion or deletion, those are very, very difficult to identify for the repair mechanisms. So many of those will be skipped and will therefore be propagated by DNA replication as a mutation, as a change in the DNA. Fortunately, again, there are ways of monitoring the DNA and recognizing if the DNA is becoming damaged significantly. And then the cell will enter a process called apoptosis, programmed cell death, which we talked about back when we were talking about mitosis and cancer. So it's basically apoptosis, and the tumor suppressor genes, they are monitoring damage to the DNA. And when they detect significant damage to the DNA, that's when they trigger apoptosis and program cell death. Oh, I probably should comment about this last one and put a note in here. Mutation is the source of all alleles, right? The flavors of genes, every flavor is actually a different sequence of nucleotides. So they are the product of mutations. And the only source of new alleles. So as much as mutation may seem like a bad thing, in fact, without naturally occurring mutation, not the mutations that are the product of things that we do, the chemicals that we make, the things that we do that we know we should know what to do, but naturally occurring mutations are the source of the alleles that natural selection requires to allow organisms to adapt and to evolve to new situations. So mutations are kind of a two-edged sword. There are pros and cons. The pro is, of course, they're the source of all alleles, and particularly the new alleles that natural selection is going to work with. But if the mutations are too significant or in the wrong location, they don't become beneficial, they become harmful. Fortunately, most harmful mutations are eliminated by natural selection pretty quickly. And it is more common for harmful mutations than beneficial mutations. But when beneficial mutations occur, natural selection can take advantage of that. So one more concept check, obviously not a break, because we are at the end of the presentation, the end of the chapter. So 2 concept check questions. What would happen if a mutation changed the start codon to some other codon? What happens when one nucleotide is lost from the middle of the gene? Well, if the start codon is altered, the ribosomes are not going to be able to identify the beginning of the gene sequence, which means they will not assemble around the messenger RNA, and translation will not occur, which means no protein. So we could put here no polypeptide. So that could be quite serious. What happens when one nucleotide is lost from the middle of the gene? So the reading frame is altered. Which is always referred to as a frame shift. I didn't put that in earlier, but this is called a frame shift mutation because we're altering the reading frame. We're moving everything sideways to change the reading frame. So that's why it's called a frame shift mutation. And the amino acid sequence will be altered from the site of the mutation onwards. So that would be a big issue because it's going to result in perhaps a non-functional polypeptide. It's certainly not going to result in the polypeptide that was intended in the original gene. Okay, so that brings us to the end of this presentation. We've covered a lot of ground. We've talked about the structure of the DNA molecule, how the DNA molecule is replicated, And then we've talked about how you can extract information out of the DNA molecule through the combined processes of transcription and translation. And then finally, we've talked about what happens when you get an alteration in the nucleotide sequence, what we refer to as a mutation. So as always, if there are any comments, concerns, or questions relating to anything that I've covered in this chapter, by all means, reach out to me in office hours or send me an email. Let's get whatever questions you have answered and any misconceptions or misunderstandings clarified before you move on to the exam, which, as I said, is coming up in about a week and a half or thereabouts, covering these chapters. So time is progressing really quickly. We are rapidly approaching the end of the semester. So when we come back again for the next presentation, we will be looking at chapter 11, and that is all about how we regulate gene expression. And there are a number of levels of regulation, some of which I've hinted at already in this presentation, but we'll look at them in more detail in that presentation. Till then, I'll see you around.